Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova. I'm pleased to be joined by A Rational Empiricist, also known as Andrew. And so we're going to talk science. Andrew, could you say something? Because I'm going to try to test the audio here.
Yeah, how's the audio right now? Is it any better? Am I coming through very well, or?
Okay. Yeah, it sounds good on YouTube. I was just double checking if I sounded a little, if I wasn't talking very quickly. So I'm going to have to listen. For some reason, the audio on my computer suddenly got very soft. So I'm going to have to listen very carefully to hear you. So you wanted to talk about evolution and phylogeny and something about science?
Right, yeah, or I didn't really have a specific topic. I just kind of wanted to hear your thoughts on your position on certain topics within what some people would call evolution, but of course, others would call it just astronomy and things like that.
Yes.
Go ahead.
Unlike some of the other YouTube creationists, I actually did study a little bit of evolutionary biology in graduate school when I was taking bioinformatics. And so I knew some of the methods. I also think that creationists don't respect the theory enough in some sense. Meaning, I've been dismissive myself, but I have immutable. No, not right now, because I promised Andrew I'd do a one-on-one with him. I could do a one-on-one with you. So, so if you'd like to immutable, we could have a discussion sometime. Because I respect you a lot. So, unless Andrew, you wanted to, if you'd like immutable in, I can invite him.
Maybe we could do one-on-one for a little bit, and then we can see if any other people want to join, and we can just have a short little open discussion. If up to you, really.
Okay. Yeah, because I have such great respect for immutable destiny, and I have, I don't want to I don't want to just outright diss him.
Right.
There was a little bit of there was actually a big mistake on my part. I actually thought that the Standing for Truth show was going to be at 4 p.m. And so I said, 8 p.m. should be fine. And I totally messed that up. So didn't mean to have a scheduling conflict, but thankfully people can watch things via recording. I felt Andrew reaching out to me was important enough. I did want to have this chat. He seemed very interested in hearing the other side. So where I was, I said I actually do have training in evolutionary biology at a variety of levels. Actually, I got a letter this year from an editor of a major publisher that something I co-authored has been accepted for publication. I can't talk about it in too much detail because I don't want to get deplatformed. I mean, it's still possible it could get retracted at the last minute. They figure out who I am. Right. So, which would really suck. I mean, I could have 3 publications this year. So I just wanted to point that out for the viewers. So I've actually studied some of this, the population genetics and professionally. Now, with like a lot of other evolutionary things, everyone has their specialty. There are people like Dapper Dino. He really knows dinosaurs really well.
Right, paleontology stuff, yeah.
Yeah. I'm not that kind of. That's not my specialty. Mine has been in population genetics and evolutionary theory, and I also studied aspects of eukaryotic evolution and then specific proteins, because it's very hard to be like a generalist on all proteins. There's really no one who's any good at all proteins because
Right.
each protein is really difficult to understand. Even something as that may be several hundred residues, amino acids. It's still the operation and its structure. And then seeing it across species is not trivial. So
Right. It's kind of like examining a new mutation in a population. It takes a long time, like years, decades maybe, to like figure out all the details of it.
exactly. So let me show some of my. Um, oh well, this. Did you ever see my show with Erica?
Uh, I saw bits and pieces of it. I know you guys have done some phylogenetics uh together.
Okay, so why don't I just to review for the viewers? I did do a show. Uh, Erica and I just mutually agreed. I said, "Hey, Erica, let's do a show on phylogenetics because maybe you could show me the way you do it, and then I'll show you the way that I've learned it." And it was actually a
Right.
very good. Uh, he was, it was a very good exchange because we learned something from each other in the
Sure.
process. Because I was actually shocked. It's like, oh wow, you totally do this a different way than I did, and and um, and then she saw that I did it differently than she did. So, um, tell me about what you know about. Have you done phylogenetics at the molecular level?
Well, actually, I'm kind of finishing up my senior year right now. I'm not undergraduate or graduate or anything like that yet. Um, so I don't know the details of it, but from what I do know, um, I guess what exactly are you asking for? Because like, or I kind
I'm
kind
just
of
trying to
something
know
like
where
that.
your, I'm just trying to know what your knowledge base is. I'm actually not very. Most of what I know about where you are is from my my brief interactions with you. Actually,
Hmm. Yeah, yeah. I'm finishing up my currently finishing up my senior year of high school. Actually, so I don't have any training on this subject, but it does fascinate me, and I do know there are many methods involved. I know the basics of um like there's parsimony, for instance, or maximum parsimony to be specific, and maximum likelihood and Bayesian techniques. Um, and there's also distance methods like neighbor joining. Um, and so I know the basics of those fields and how they're classified basically. So I guess more so what it means to be phylogenetically related rather than how they're constructed is kind of like I know a little bit of both, but what one's a bit more biased than the other in terms of what I know.
oh yeah, no, it's just so that we're talking the same. I just want to know where, um, uh, kind of where we are, so we can just communicate
Right,
better.
right.
So
Yeah,
I
yeah,
think
I
it's
understand.
outstanding that you're looking into this at your level of education. Are you planning to study biology soon in college or?
Yeah, actually, I'm hoping to major in evolutionary biology if
Okay,
when
all
I
right.
enter college.
That's uh uh. I have my opinions about evolutionary biology, but you'll be learning a lot of biochemistry and
Right,
other things in science, which I think is great. Um,
right. Yeah, I know we disagree on this subject, or at least parts of it, but like I still appreciate you letting us have this conversation.
okay, so part of the reason I asked, I'm just gonna also partly because I had this presentation I had for Erica,
Okay.
and I'm just gonna maybe just present it. Uh, so if it seems like it's like, why am I talking about this in a discussion with you? I just happen to have a slideshow that I happen to have done with her.
Right.
And so if you see some slides and they seem kind of like irrelevant, that's the reason. But so for the viewers, um, I was just trying to see where you are. And then also, I'm just going to talk to the viewers a little bit too as I go through this. So I'm really
Right.
glad you brought up the topic of phylogenetics. So let me see. Let me just look quickly through my own slideshow first. And I'll give
And
also a little
if
bit.
I may clarify something, so I believe we've talked before about how you're okay with phylogenetic methods, but you don't think the three domains of life are related, correct? Is that a good summarization of it?
Um, hang on, I'm creating a few problems. There are a couple technical problems here from that I created myself.
Oh, sorry.
Uh, so if you see me bending down like that, I'm having some strange problem with being able to hear. And let me see if I can. I'm going to try to put a. Andrew, can you talk?
How's this?
Oh goodness, I can hear you better.
Oh, cool.
Thank you.
You're welcome.
I had a major. Let me. Yeah, boy, I can hear you way better now.
Okay, that's good.
All right. I don't know what's wrong with my poor. I apparently blew up my microphone. Okay. I'm just looking at something I did for Erica. If you'll give me a moment.
Also watching the chat as you're going through that.
Yeah. Okay. So can you this is going to help our communication better. You asked me something about phylogenetics earlier,
Right.
and I it got garbled, and that's why I had to suddenly try to grab my headset. So what was the issue?
Oh, so from our previous chats, I'm guessing, or I presume you accept phylogenetic methods, but don't think it goes all the way to the root of the three domains of life. Is that a good summarization of it?
Um, Not just the three domains of life. Okay. So what I'd say personally, uh, I don't think that we evolved. Humans and primates evolved from a common ancestor because I don't think there was enough time. So if one accepts the young Earth model, then there's just no time. The similarities then would be by design. If one accepts a old Earth model, one could say, okay, I might like Dr. B. He accepts intelligent design, but he doesn't think that, uh, and he accepts universal common ancestry, but he thinks there has to be intelligent design along the way. Now he doesn't use the word miracle, but um, that's the way I would interpret it.
Right.
Is that there are these gaps? So so you may hear me say, okay, if we accept this model, this is not feasible. If we accept this model, this becomes feasible, but then there's still some issues there. So I'm expressing my personal views, and then I'm going to also say what I think is and is not consistent with a particular model.
Right.
And then, of course, the question is which model, particularly of the fossil record or the age of the Earth, is valid. So, and I'm happy to acknowledge, let's work within the models because
Right.
I think we could still learn something and understand some aspects of things, even if we accept certain models just for the sake of argument that we may not personally accept. And
Right.
we,
Yeah.
we, we,
Oh, sorry.
oh, go on.
Or my personal way of looking at it is that in order to learn, you have to know what people already know, if that makes sense. Like what the current standard is, I guess.
Well, even in mathematics, there's one of the most famous proofs was the square root of 2 is irrational. And it was very hard to actually solve that problem. So what they had to do is they say, let's assume for the sake of argument that it is rational. And once they did that, they were able to do what they do called the proof by contradiction. and so having different models and trying to work within them, we do actually learn something, even if
Right.
it's not the model we're promoting. We can start with a model that's different than the one with, that we might favor. And that was a very good example of it, the square root of 2 and its irrationality. So I'm going to show a little bit. What I will do, let me see if I could find the link to my talk with Erica just for the benefit of the viewers.
Okay.
And I'll post it in the side chat. Let's see. Hang on. I can't do it on this computer. It's one of the bad things about both StreamYard and Restream is it's very hard to it's hard to post on the side chat on the same computer at the same time.
Right.
But I'm going to look for my talk with Erica. So I have a really slow computer here. But in the meantime, let me see if I could share my screen.
Okay.
And I'm going to look at,
Oh, nice.
I'm going to look for that video I had with Erica. And let's see. Okay, I found it. Yeah, that was a real fun show for both of us because most of the time you've probably seen how other creationists have interacted with her, and
Right.
I really didn't like that level of hostility personally.
Right. Yeah. I mean, it's what I think is, yeah. Or sorry, I lost my train of thought for a sec. Continue.
So I'm gonna.
I'm trying to
I
think.
have a really slow computer on the other. Okay, so I found it. So this is my show with Erica. Okay, I just typed it in the side chat there for future reference, and you'll actually see it. But I'm going to go through. I'm going to go through that talk. So this is my presentation on molecular phylogenies, and so this is one way that the one reason it's nice to do molecular phylogenies is that because of sequences, we can also do things computationally. Because if we're looking at bones and morphology, it's like, yeah, does this fit here or there? This is much more repeatable. We could say it's objective in the sense we know what parameters are there.
Right.
Okay, so why would creationists study molecular phylogenetics? It's in my case, it was because the boss told me to study it. He sent me to the NIH. I had to learn it. Otherwise, I probably wouldn't have been interested. But also, it was for a project we were working on, and I'm going to highlight the details of that project. There are creationists that study it for the sake of baraminology or the dating of Adam and Eve. I'm not that interested in baraminology. I am, in passing, interested in Adam and Eve, but that wasn't my professional specialty. So
Okay.
those of you who know me know that I worked for Dr. John Sanford for 7 years. We recently completed the contract, 7-year contract, and it was a real privilege to work for him. And part of my job was to learn more about whatever I could at the NIH, given the time that we had. I had to, so some of what motivated me getting assigned to learn some of this, the primary reason was there was a 1984 hypothesis that was by Susumu Ono, and it kind of, I was tasked to try to falsify it because Dr. Sanford felt that there was something wrong in the papers that got published in the proceedings of the National Academy of Sciences. And if we could do that, we would kind of cast Ken Miller, who was in an infamous 1984 trial, Kitzmiller versus Dover, and also Dennis Venema and the National Center for Science Education. And we would look to see if there's anything that could be done that might quietly acknowledge the lack of a universal common ancestor between of proteins and genes. And there was some citations here: Honig and Petrie, Wolf and Kunin. And then it turns out we might be able to use some phylogeny tools that, even though we don't believe in the phylogeny, we can use it to inform structural biology and protein science. This is really advanced stuff, by the way. This is like so cutting edge, you won't find it in textbooks. And this is stuff that I'm actually I'm hoping to be published on hopefully very soon. So we got some letters from the editor, and I can't take credit for it because my co-authors are the brilliant guys. They're actually structural biologists and enzymologists and computational guys, and I'm just like a data clerk, and I just had the privilege of being a co-author. So this is a little bit of background. I was this gentleman here is Eugene Kunin. He's the head of the evolutionary genomics group at the National Institutes of Health, the department called the NCBI, National Center for Biotechnology Information, or something like that.
Right.
Let me see if I actually identified my professor here. No, I'll tell you who my professor was. One moment. My professor here is in the orange. So he's Kunin's staff worker, and he was my professor at the NIH. By the way, I don't know if you've heard of Cy Gart. He shows up in YouTube circles. He actually was a professor at my school also. And the book that was supplementary reading was this one by Joe Felsenstein, Inferring Phylogenies. And this does it at the computational level. Now, I mentioned earlier about some of the issues with looking for a universal common ancestor among proteins and genes, and it's related to this paper as Protein Classification Necessary by Honig and Petrie, and also Kunin and Wolf. They're talking about the lack of universal common ancestor among proteins and genes. Now, that's not the same thing as the universal common ancestor of organisms. And what I found in like the last decade or so, that the idea of a universal common ancestor in terms of organisms has been kind of, it's ambiguous now. They're talking about a universal common ancestral population. So when you're talking about the domains of life, that's now, even by evolutionary literature, they're like, no, it's not from the same single cell. Maybe it was a pool of cells. I mean, this idea has been floating around since the time of Carl Woese and Doolittle. And I'm just throwing that out there.
Right.
So this was the famous lawsuit. It involved Tammy Kitzmiller. She was suing the Dover school board that were a bunch of creationists who lied, which, I actually sided with the evolutionists in this case for getting after them because the creationists lied, and that's just wrong. That puts people like me in a very bad light because of their behavior, puts shame on everyone, and I just thought that was wrong. And it resulted in this trial. She was suing this school board for promoting intelligent design, and that was just a total mess. But there's this biologist here by the name of Ken Miller.
Right.
And he's a professor at Brown University. He was called in as an expert witness. So how did this involve me? Well, he was advocating a certain theory that also Dennis Venema in 2016 advocated. And in fact, this particular claim, it was his favorite claim about protein evolution. And I said, okay, this is going to be a nice big fish if we could show that the basis of what they're promoting is wrong. And this will lead to why I was interested in phylogenetics. I might be able to even see if I have it here. Yeah, I have the mega program going. So hang on. So I'll just proceed here. And he said evidence of evolution and how new proteins can evolve is based on this. Let me see if I could find it. It's based on. Sorry, I'll talk about it specifically. I have the paper in mind. But both Ken Miller and Dennis Venemus cited a paper by Ono, and it was also cited in 1985 by the NCSE. And they wrote this whole big thing, the new proteins without God's help. And they're, because the creationists will argue for the improbability of evolutions. So we wrote this paper. It got rejected by several editors. We asked the editors to release, if it'd be okay if we could release the rejection letter. They would not. And there are a number of maybe good or bad reasons for that, but that's just the way it was. It turned out the editor-in-chief of Cell Reports looked at our paper in passing, and he didn't say it outright, but he agreed with our conclusions. He said Ono was wrong. Ono was wrong. Joshua Swamidas agreed with us that Ono was wrong. So Joshua Swamidas agreed with the conclusions of our paper. Steve Matheson, editor-in-chief of Cell Reports, and also John Harshman, PhD evolutionary biologist. I've provided a link right here if you want to look at it, if it's still up there. Hopefully Joshua Swamidas has not taken it down now that I've said some really negative things about him recently. So we got all sorts of reasons why our paper was rejected. It wasn't because our conclusion was wrong. We got all sorts of creative reasons, just suffice to say that. Usually on the order, it's like, why are you publishing this? This is long falsified, something like that. Or it's not novel, or it's out of the scope of our paper. But we really did want them to give us specifics and then to let us share it, but they didn't want to do that. And I'd also say this, it would have also put the whole industry kind of in a bad light. It's like, guys, why aren't you correcting this? My issue was I didn't at that point care whether our paper got published or not. I wanted the editors to come out and say, hey, this has never been corrected. This paper is wrong in the Proceedings of the National Academy. It actually was also wrong in Nature 1983. So it was kind of frustrating. So just briefly, because I may talk about this, this is one of the major proteins that I study. This is the topoisomerase protein. It cuts DNA to untangle it, so it has to cut it and then actually stitch it back together. And the reason you have the blue and the red. Oh, by the way, one reason I asked about your background, Andrew, I actually thought you were a college senior. You were so smart and knowledgeable. I thought you were a college senior already. So I'm doubly glad I asked now what your background was, because you seemed really knowledgeable. So
thank you.
For the viewers' benefit, this is a depiction of the topoisomerase. It is the reason you have the two colors here is the gene has to express two copies of the topoisomerase 2 alpha polypeptide or protein, and then the two copies will then join together to make a functional complex. Unless it does that, it's not going to work. And that's going to be significant in the discussion of phylogenetics. That's one of the scientists I've worked with. He's a young earth creationist. He's a adjunct professor at Vanderbilt School of Medicine and also associate professor at Lipscomb University in biochemistry. So there he is. That's Joe DeWeese. He published in the prestigious scientific journal Nature. Let's see if maybe a little bit of bragging rights here. I actually was in the prestigious scientific journal Nature in 2005, April 27th, just for the reader's benefit. The cover story, Who Has Designs on Your Student's Mind? You'll see me in that cover story. So, just to say, yeah, A Young Earth Creationist got published in a major scientific journal on the topoisomerase protein. And then both of us got published on this in 2019. Unfortunately, it had to be April 1st. It's like, no, that wasn't an April Fool's joke. That was for real. So this is the paper on that was in question. They said this is this was cited for a long time as an example of de novo protein evolution. They said it happened by frame shift. Are you familiar with frame shift mutations?
Yeah, like insertions and deletions and stuff like that.
Yes, you have
It
almost
shifts the reading frame basically of the codons.
right. Very good. See, that's why I thought you were a college senior. You're so smart. And so what he did is he analyzed it. He said, oh, well, maybe if it were just slightly different by one frame shift and then it got shifted, we'd make this brand new protein. I mean, it was a good try for the time, but are you familiar with BLAST or BLAST-P, these particular tools and websites?
I've heard them. I haven't looked into them, unfortunately.
Okay. No, this is good. This is good because actually I was actually surprised that wasn't her specialty, so I had a chance to show Erica this, and she said that's really cool, and I'm like, well, that's great because then, we actually had a productive discussion then because
right.
I was saying I wanted to find out how you do your phylogenies, and I'll show. So let me, for the reader's benefit, then for the viewers, since we're talking this, and it depends, I guess, on how the curriculums are structured, how, to what extent this is going to be taught in various schools. And like Erica's a biology major, and just by the way she was reacting, it's like, no, that wasn't anything that they focused on. It could be just the availability, what the professor had a preference for as far as tools. So I'm not,
right.
I'm not knocking her. There's so many tools out there.
Oh yeah.
So, I go to this website called Uniprot, and I happen to know the name of the protein, the topoisomerase. It's known by the identifier TOP2A. And so I just go there and I type it in. And then we get all this listing of all the creatures that have TOP2A.
Okay, I might actually know this website. I've heard of all of them, but like I might have actually looked at this one if on occasion.
Yeah. Well, most, definitely a lot of creationists don't know this. So, so this is the actual amino acid spelling of the topoisomerase protein.
Okay.
And there was somewhere here, I think it's around 1,500 amino acids. It's in that ballpark. So that's all it is.
I'm guessing each amino acid has a particular letter.
Yes. So there, it's the alphabet is about 20. Now we have these other amino acids like oh gosh, I can't remember it, but
yeah,
so yes. So this is the M is for methionine, P is for proline, L is for
leucine.
Leucine, right? Immutable might know all these. N is for asparagine, I think. V is for valine, S is for serine, R is for arginine, K is for lysine because they ran out of letters. Y is for tyrosine, G is for glycine, I is for isoleucine, A is for alanine. I'm trying to remember that. So this is leading. So this is how what we would do then is I would collect all of these letters, and let me see if I happen to have this.
Okay.
If I think I happen to have a supplemental file here. See if I'll pull it up. Okay. I can also, the protein in question was NYLB. So this is what we did our rejected paper on. It, only 392 amino acids.
Hmm.
And this, I mean, this was a major, there was a lot of research just done on this right there, right on that paragraph
Hmm.
size. That's hardly even a sentence. That's not much more than 2 side chats right there. And there was a lot of years of research on this. So,
Yeah, or definitely a small protein.
yeah, actually, that's about the average size. I heard PZ Meyer says he believes the average size is about 430 amino acids for the proteins in the databases he had. 430. Now the
Hmm.
eukaryotes can have some gigantic ones, but I don't know the methodology. But
Yeah.
anyway, what we
Then
did is
again, what does average mean anyway when there's so much variation?
yeah. So what we did is we collected all of the there are lots of NILB proteins scattered across all these species, and I collected them. And I did a phylogenetic analysis on it. So let me see if I did that. So what I did, let me go back here. What I did is the first thing was I went to Uniprot and I found it. And then once I got this sequence here, once I got the first sequence, let me see if I can, let me bring this down, Uniprot. So I found this sequence here on Uniprot. Then I went to Blast, and I'll show you the Blast website. So let me
Okay.
go there. And I'll just show you how this works.
Sure.
Because this, I just wanted to demonstrate to you that I and also to the readers that that I really do have, respect for what they're doing, and
Right.
it's also for the creationists out there. It's like I want them to be maybe a little bit more circumspect in their criticisms, because
Right.
once
Yeah,
because
we
I've
can
been
get
trying to
into that
tell.
later in the conversation.
Yeah, because it's like, guys, the way you're talking, it's like if you did a little bit of protein analysis, you might not be saying some of the stuff you're saying, and so it's um, I'm interested in having creationists represent our case correctly. So what I did is I took that sequence that I found, and I said I can go to the Blast website, take that sequence, and I could present it and say, go find as many sequences as you can that look similar to this.
Hmm.
And it'll take it about several minutes to do this. And this is what I showed Erica. She said, wow, that's really cool. I
And
mean,
and this is from like what the like it searches current literature basically.
uh, it, uh, it, it searches a database. So what happens is
Database, yeah.
they have several databases. One is called curate. Some are what they call curated, meaning they have to have really high standards for it to be in the database. And then they have uncurated databases where laboratories will just report. They'll do a gene sequence on their. Let me pause a little bit here because see if I could. They'll do it. they'll do gene sequences of the genes, say in the organism they're studying, and then they do what's called a protein prediction. They don't actually. It's very actually hard to extract proteins from a creature and then actually look at the individual amino acids one at a time, because that often involves like mass spectrometry or spectrometry. It's a really expensive process. So
Right.
what's easier is they'll sequence the genome, and then they'll estimate what proteins are generated. It's not 100%, but it's
Right.
cheaper. It's actually pretty hard to I mean, people, it can be done, but you have to be really determined if you want to get the exact sequence.
Sure.
That might be done if they're trying to determine what are the alternative splices or any post translational editing, which does happen. So yeah, so Immutable Destiny weighed in. So thank you. Yeah, it's not just suffice to say it's financially expensive to actually do that. So the reason
Right.
I was pointing that out, you asked where this came from. So they'll just deposit the results of their experiments. The quality of their experiment is just accepted at face value. If it's been, I think they usually like to have a report whether you did get it peer reviewed when you published your sequence. At least that's the way it used to be. I don't know now if the labs will just, if the databases will just accept it. And that's actually good, because why would anyone report a bad sequence? It's just not cool.
Right.
And so they'll dump this, and then yeah, there could be mistakes, and then but they'll dump it. They'll also provide their estimated protein sequence that they think might be translated. And then so in this BLAST-P, what ends up happening is they'll have all these protein sequences. BLAST-P will search for ones based for similarity, and it's actually shocking how actually dissimilar they can be, but yet they can find enough similarity that's above that would be considered non-random. So, goodness, my search here is still going. So it's probably searching millions
Oh wow.
of sequences. Maybe tens of millions would not be an understatement. So it's going to look for similarity. So let me just then proceed with where I was. While this job is grinding away. By the way, you need a little luck in advance to know what the identifier is to go to Uniprot in the first place, and that's also a pain. So sometimes that's why it's good to be able to be a part of a community where you can talk to someone and say, tell me how to look for this, because I have no clue, because their paper may describe kind of a colloquial name for it, but then they don't tell you exactly what identifier. It can be obnoxious. And it was I'll tell you how I did it. Actually, when I started, I painfully typed in this entire sequence from
Oh wow.
hand. Then I went to BLAST-N and put it in, which is the nucleotide sequence. And that's how I found the identifiers. That was not pleasant, because if you don't know the original researchers or the people involved, that's kind of what's unfortunate. You do have to have some. It helps to have connections. There's no question about that. So, let me see if I had some pictures here. Do I have pictures in our paper? If you'll give me a moment, I'm going to see if I could find. I'm trying to look for. I happen to have a picture where. Oh, I know what I did. So, let me see if I could do this. I have this. Yeah, what I did was this was a live demonstration that I did with on Erica's channel.
Hmm.
Because what I'm trying to, one reason I'm going through this is I want to convey to creationists how powerful, how the nested hierarchy is very evident, and I don't think they appreciate it.
Right.
So
Oh, you
this
got
is
Mega up there.
yes, yes. Or are you familiar with it?
I actually tried playing around with it recently, but I couldn't. It seemed to take up a lot of space, and I couldn't exactly figure out how to get into it that much. So
Okay. So let me see. This is kind of, I kind of
I'm
joked
not
with
much of
Erica.
a
I said, you
techie.
never want to do a live demonstration because it could blow up in your face.
Oh,
Uh, but I'm going to go ahead and try. Let's see. I'm going to try doing an alignment. So do you remember? Let me see. I had all of these sequences that I got um, that are similar. So this was from a blast, the actual blast run that I did.
right,
And I picked these out because I felt they were representative. So these are from 10 organisms here. And then, so now when I put it in Mega, um, so I mean, if you want to try it with my sequence, this would be a good exercise. You could actually just type, I could give you this file, and this is the way it needs to be formatted in the FASTA file. And so you just take the protein sequences, and you have to put some spacing in there, and it'll identify the various protein sequences in these organisms. And then what I did was I did a file open. Why don't I just start this from scratch again? Just so when you open Mega, you get a screen like this.
right. Yeah, I think I remember how to uh enter it, but you can show it just in case.
Yeah. So then you have to have your FASTA file, and I
Right.
just do file open, and you open the FASTA file. Say, do you want to align? And so you say yes, and then uh, I use the muscle alignment algorithm, and it's going to try to get everything lined up. And there are kind of nice. I use the defaults here just for starters. The
Right.
aficionados will know it's good, and then it grinds away. And then I say, just to highlight I'll toggle the conserve sites. And then I want to use, yeah. So anything
Oh,
that's
oh,
100%
Emery is here. Cool.
Oh, hi, Emery.
May or if you want to let someone in later for open discussion, I'd be okay with talking to you and Emery.
Oh, that'd be great. So then this is actually what's kind of shocking is
Anyways, yeah,
they're only specific. You see how many?
yeah. There's a lot of conserved regions.
Well, there are also a lot of non-conserved regions, but it's
Right.
able to identify it, which is amazing. It's only about 5%,
Five
but
percent of what?
of the 392 amino acids,
Oh wow.
and that's you could see it here. These are all the 10 creatures, the
Okay.
10 bacteria, and how it implements it. So you could see there's variation, tremendous variation, but then you have these regions where it's 100%. When you do enough of this, you'll see where there's some proteins that are like the histones, they're almost 100% conserved, every amino acid across the mammalian species. It's kind of shocking. And then you
Hmm.
look at something like this, it's only 5%, where you get this column all colored in like that.
Well, as many people have said, nature is under no obligation to be simple.
Yeah, yeah. So but it's amazing that the computer could figure that, hey,
Oh yeah.
this is similar enough that we can do this. And let me see, there's a critical, it's this pattern here, v the S, and then two amino acids, and then a K. where that was headed was on my, you see the S and the K here? They're very, very close together. And it turned out those are really, really important in the chemical action of this enzyme.
Hmm.
And where I was headed with this is Ono said this protein emerged all of the sudden after 1935, after the invention of nylon, because this S and K were very critical and it could break apart nylon, a man-made product.
Right.
And he said, well, since nylon was invented in 1935, this had to be an evolutionary novelty, which turned out to be wrong. The breakthrough in our papers, we were able to establish that this wasn't an evolutionary novelty, because mammals actually also have the capability to break down nylon. There was an experiment conducted in the 1950s. They didn't use the word nylon. They used the word aminocaproic acid, 6-aminohexanoate caproic acid.
Hmm.
And so you can imagine how miserable it was to find that experiment, because it was in the 1950s,
Right.
and to dig that up and to find it under a different name, that was not easy. But we were able to establish that mammals also had the ability, and it's just like now, mammals didn't evolve the ability to break down nylon after 1935. It would be ancient, and that was the breakthrough in our research. And now when I ran Mega, so just here, we're here on Mega, I can do, let's see, can I save this? Export the alignment into Mega format, which I can do. And
Now some of this is
I'll just
starting to
call
come
this
back to me.
demo. Yeah. So I say demo. I mean, that's great. I mean, I hope you're having fun with this part. So I exported it. And so now I ought to be able to, I want to build a phylogeny.
Right.
And so now I could do, like, say, I like the UPGMA tree, not for any good reason, okay? Just because it's cute. That's all.
Right. It's simple.
And I'm just going to run and I can make a tree. So I'm trying to show the
And for
creationist.
the, oh, sorry.
Go ahead, Andrew.
Or just for clarification for the viewers, would you mind like explaining UPGMA and how it's a distance-based method instead of something like parsimony, which is based on I don't want to say like individual characters, but yeah, am I saying it correctly or?
There are technical differences enough that I couldn't do the topic justice. So
Okay.
I don't know would be more straightforward way to say. I don't know. There is some subtlety here where UPGMA, it's a very quick calculation.
Right.
And Joe Felsenstein's book goes into the differences. This ends up what they call creating an unrooted tree, meaning they'll just assemble them and try to get them as close in terms of similarity as possible.
Right.
So we could see here, like, for example, these two proteins and these two different creatures are very, very similar. And then just based on the distance, just the visual distance, you could see how different this one would be. Very different from this one. But this one, this SME 458, is very similar to this one. So they're grouped together, and there's some sort of algorithm that tries to build a tree of similarity. So I tried to say to creationists, you're going to, they're just a little bit too dismissive of these nested hierarchies. You're going to see them if you do gene browsing. And
Right.
and the reason I said this is important, you can't just be telling these high school kids, oh, evolutionary is so bad, and then they go to college and they actually start doing the sequencing, looking at sequences and then running it through software. They're going to be like, oh my goodness, I've been lied to. Someone said that there's no nested hierarchy. You can clearly see that there are these nested hierarchies of similarity. This is like a family tree.
Right.
And
I mean, if you're gonna make a claim, you should at least put some evidence forward.
yeah. And I was like, oh my goodness, I've had colleagues say that there's no nested hierarchy. Said you can't say stuff like that. Don't
Right.
say. Now we could take different proteins and you can build different nested hierarchies, and that'll be problematic for evolution. But you can't just say that there's none. All right.
Right.
Don't say stuff like that. You'll get yourself in trouble. The other thing is when I've done chimp and human proteins, you'll definitely see that the chimps and humans will frequently align. In fact, they'll be 100% similar in some cases, but you almost always see them grouped together. It's rare that they won't be,
Right.
or very close. And you can't run away from that. So I said, if you're going to criticize evolution, you better recognize this is what um, a biology student will have to face, and you have to account for it. So
Or
in any case,
oh,
go on.
or just a question I might ask would be: would you agree? Would you personally agree that um, probably not all, but many of these trees are statistically consistent with one another?
Um, they are often, but not exclusively. And there's it's statistically similar enough that I could see why people would accept universal common ancestry, even ID proponents like Michael Behe.
Right. Okay.
It's significant enough, and the creationists have to have an answer for it. I actually think I and others have found it, but we haven't articulated it well, and that's the subject of. I might, if I have time, I might actually cover that a little bit. But we can't
Okay.
run away from this nested hierarchy. This is
Right.
a very strong argument for evolution. Okay, so even if a theory might ultimately be wrong, it doesn't mean it doesn't have good arguments in its favor. This is a
Right.
very strong argument in its
If
favor.
anything, it would be incomplete, right?
Um,
Like not necessarily. Maybe some aspects of it are wrong, for instance, and some are accurate. But like maybe bits and maybe there's a deeper theory waiting to be discovered.
I'll grant that as a hypothesis, and
Okay. Sure.
yeah, and I would find that respectable to even acknowledge where there are challenges, and that's what I would actually argue. I'm not, when I try to tell the students, okay, I'm just going to level with you. This is where I think there's an inconsistency, and this is where it's actually a very strong argument.
Right.
And what I'll tell creationists is, I said, the software you saw me use just now, the creationists used it to do mitochondrial Eve studies and atom Y chromosomal. So don't say this is bad software. That's, I always get that implication that this is bad methods. I'm just like, no, because the baraminologists use it.
Right.
The Nathaniel Jeanson uses it. Robert Carter uses it of CMI. Nathaniel Jeanson of AIG uses it. So what I just used now is exactly the same software that they use, but they don't think that this. They think at one level, it, like I do, that it is because of common descent from, like, say, a common Adam and Eve. But then the other similarities they'll say by common design. It's like, well, you could just say that, but on what scientific basis can you not universally apply it?
Right. Now, I guess if we want to clarify this to the audience, do you think common design is a universal, or do you think either are universal explanations, or do you think one has at least a better job of explaining most of the data?
It depends on what model. If I've said, if life is young, I would, I mean, I've posed this to evolutionary biologists. Sometimes they just They don't give a direct answer. I don't remember too many direct answers, but if let's just say if the Earth is young, then
Okay.
it would be common design.
Right.
So under that model, that's why there's a lot invested in it. But just to say, oh well, uh, I'll just assume it's common design, that's not very convincing. You have to
Right.
have some reason for putting a model that will actually rule it out, not just because you read it in the scripture. I mean, that's perfectly fine if you accept it by faith. Um, if you believe that, um, as I do and others do, that God's word was divinely inspired, that's legitimate. But you know, it's like you need some evidence that would persuade you that God's word is inspired.
Right. And I,
I
I,
don't think
I
it's
think, oh, go ahead. Sorry. Didn't
yeah.
mean to interrupt.
So I mean, just give an example. Let's say someone was miraculously healed in the name of
Okay.
Jesus. I would consider them, if they start accepting God's word from that point, I would totally respect that. But you know, for someone that doesn't know anything and they just trust, it's like, okay, that's great for them, but it's not very persuasive to outsiders. Okay.
Right.
um I mean, you know, so uh I think it's, I mean, as a Christian, I think it's great someone trusts God's word. But if they're going to try to persuade other people, it has to be more than, well, I trust God's word. And even as an insider, it's like, well, I'd like it to be a little bit more. Can you give me some evidence? And so I was, um, you know, so I would say, when you ask me about the statistical consistency, is it really strong? It's like, it is extremely strong.
Right.
And I don't know that it's easily refuted. So that's why we would have people like Michael Behe, who, even though uh, he accepts intelligent design, he's also willing to accept universal common ancestry.
Right.
But he would say that there needs to be an intelligent influence to make this happen. So I wouldn't just say, oh, it's common design, just because I think, oh, well, it could be. You
Right.
can't.
It always can be. It's
Yeah,
always
it always
possible.
can be. You have to give a good reason
Right.
as to why it's highly probable.
Right.
So that's why the young Earth model is, I would say, would be the best way for people who are arguing for common design to prosecute their case. Otherwise, you know, if I didn't have some strong suspicion that the young Earth model was correct, I'd probably fall back on Michael Behe's model,
Right.
because that would actually agree. It's like, okay, Michael Behe, outside of the young Earth model, he would represent my views in terms of, like, wow, I see this nested hierarchy, but yet there are other problems in creating de novo proteins.
Right.
So
Yeah.
speaking of which,
I
go
oh, sorry.
go
Or
go
just
on,
one last
Andrew.
thing. I think the one other thing I want to say is what you said kind of shows that you can have common descent and you can have proposed mechanisms of evolution, which I accept, but perhaps others like you won't accept them. Um, but like it comes to show that the two aren't um, like they're related, but they can be mutually exclusive. Like one doesn't inherently depend on the other, in at least in most circumstances.
Yeah, in a lot of circumstances, I think I get the drift of what you're saying.
Right. Because perhaps it's and this is the one other thing I want to say was that I think you can falsify a parsimonious creator. By parsimonious I mean, okay, we kind of know what he's going to do based on like how people design cars, and for instance, like that's the often repeated analogy that I hear, which I think isn't really up to par, but um I kind of think that that
I'd
kind
actually
of
agree with you there. I'd actually agree with you there.
okay, right,
That's why
like
I said you can't just invoke it. You need some data point that's going to force you one way or the other. You can't just say, "Well, I like that idea," which is
right.
almost, yeah.
Right, because both models are possible, but one isn't, or well, one, but both are possible, but they're both not necessarily equally likely based on the available evidence.
Right, right. And so that's where I say, people who are actually in the college, we'll hear the YouTube creationists say stuff, and it's like, have you ever actually studied this at the college level? You might not be so quick to say stuff like that,
Right.
because I know that they're going to wrestle with it. The reason I know is, even me as a creationist, it troubled me. It troubled me. I said, if it troubled me, I know it's troubling others. And I'm saying, this channel is organized to actually try to reach out to the creationists and ID proponents that are actually in biology programs or science programs at the college or graduate level, because it's a different discussion when you get there. It may be one audience might be relatively easy to persuade, like those people that listen to Kent Hovind, but it's a different audience if they're actual biology students in college.
Right.
So
Right. So just to sum up this portion of the chat before you move forward,
yeah,
we would both find common ground on the idea that you if you're going to propose design rather than common descent, like just throwing out analogies like how cars allegedly fit into nested hierarchies or stuff like that, that's you're gonna have to put more out there than that.
you're going to have to put more out.
Yeah.
And
Okay.
okay, so the easiest way that I have said is, okay, and it's really not so easy because the young Earth model is still very challenging.
Right.
We can make some isolated arguments with proteins, and you'll hear me talk about the eukaryotic prokaryotic transition, because you can actually see the nested hierarchy will not serve as a good explanation, because I've actually analyzed some of the proteins. It would be very difficult to say that common descent is a good explanation without some sort of miracle along the way. But I'm getting way ahead of myself. So that's why people say, well, why do you keep talking about that? I said, that's where it's really easy to prove that you can't, even if we assume some sort of naturalistic evolution, you would need actually miracles to make this work.
Right. And
But that
and
is way down in the weeds. Go
right.
on.
Obviously, we disagree on that part, but I think we can at least find common ground on the fact that this hierarchy exists, and there is a model that explains it currently.
There is a model of random mutation plus natural selection, but with the eukaryotic transition for the proteins that are shared across the three domains of life, it is, in my opinion, very, very serious. And
Okay.
and
Yeah, I'll have
I'm
to look
very
into that.
emphatic on that, but it really is down in the weeds. I had a show on my channel that specifically addressed this. I had biochemist, professor of biochemistry Robert Carter, and professor of molecular biology Change Tan also weighed in, and we were expressing reasons from a scientific standpoint that this is statistically, you can't just use a phylogenetic software and say, hey, this, look at our similarity diagram, this proves evolution. We did speak our conscience and say, and me just as a human being to another human being, I can't in good conscience say that we could run this through Mega and say, hey, they're evolutionary related. That would feel scientifically irresponsible. But I'm getting way ahead of myself. So I
Right.
will
Yeah.
agree
Yeah.
that you can
Go
build
ahead
these
with it.
trees. You can build these trees and show the computational similarity computationally. We can also build the tree we can render the same tree. This is actually the same tree rendered differently here. There's a certain name for this. Some people call it like a starburst, but there's probably another name for this.
Right.
Let's see if it had. There's something in Mega here where you can describe the way you want to render the tree, and you can get it to render like that.
Right. Or you can
So
move it.
where did this go? When we looked at this tree, it's like, oh my goodness, this looks like an ancient enzyme. It didn't evolve after 1935. If we even assume a standard evolutionary model, and we found it in an obscure paper that wasn't directly addressing this even in the first place. The paper had to do with stop codons and the antisense strand of the genes for nylon oligomer degradation. But they said most of the amino acid substitutions from the ancestor of NILB gene family to its descendants of today might have occurred before the beginning of nylon manufacture. And it was estimated the origin of NILB happened at least 1.4 times 10 to the 8th years. That's like 140 million years ago, prior
Hmm.
to okay, and this is my comment. So this is in the yellow is the original paper. And then I added, so that's prior to the present day, 140 million years prior to the present day. And I said, this effectively falsifies Ono's 1984 frameshift hypothesis. So in our paper, we did this, and then we also cited this. And our
Hmm.
paper still got rejected. I said, what was happening, what was bad, was Ken Miller, who was in the Dover trial. Let me see here. There's Ken Miller, Dennis Venema, and the NCSC, as far as I know, never made a retraction. And this experiment, this paper by Ono is still cited in peer-reviewed literature. And yet we have the editor of Cell saying that original paper was wrong. Joshua Swamida says it's wrong, and other evolutionary biologists. My NIH professor said it's wrong, and yet it's still being cited as a correct theory. And this is the paper in question that got falsified. So we actually got to use a phylogenetic method to falsify an evolutionary paper, and it's a paper that's still cited as correct.
Hmm.
So we falsified the NCSC claim. We falsified this paper. We falsified another paper that was published in Nature 1983. And so that was our champagne. So Erica gave me the privilege of showing this on her channel. And so I just walked you through it.
Hmm.
So where does this lead? I just wanted to show you the basis of, let me see, I'm going to stop sharing my screen, of where I'm coming from, that I totally agree with you how powerful the nested hierarchies look. And so this is, I'm going to tell the young earth creationists, you're not going to have an easy time falsifying it. Don't think that God makes this easy for you, is
Right.
what my advice is. Don't think that you can just walk over evolutionary biologists.
Right.
You
Right.
just won't have any credibility.
Like I said, I disagree with you on some points, but then again, I kind of disagree with other people on certain points, and I respect you
Oh, thank you.
actually addressing some of these points and using the evidence. I don't want to call out any names here, but I have seen some YouTube creationists who don't do that as much. So I appreciate that.
Oh, thank you. And you see, I know that God will, that there are other biology, creationist biology students that aren't hearing this. I'm just like, we don't serve the creationist community well by making them think that, by making them disrespect the arguments that are actually strong by the evolutionary biologists. I said, you guys are going to, some of these homeschool moms are going to just think, oh, we gave them answers in Genesis and Kent Hovind we'll send them
Right.
off to school, and they're going to be able to survive that environment. And just like, yeah, you don't know what you're doing, and it's not good. It's not
Right.
healthy. Now, Nathaniel Jeanson, I asked him, how did you make it through? He said, I was always given both sides.
Hmm.
I said, how come you were able to stay a creationist and get through Harvard? He said, my family taught me both, gave me access to both sides. And I could say, yeah, I could see why he was able to make it through, because he was given appropriate respect for the arguments of the other side. Because if you walk into there thinking what Kent Hovind taught you is going to be uh, enough to get through school, it's like you're going to be shocked. You're going
Right.
to be shocked, and some of them are so shocked that they'll leave the Christian faith.
Right. Like,
So
yeah, I don't think those resources are particularly helpful if you want to maintain any faith you have. So, which I can't speak on it that much because I'm agnostic on the issue, but I get what you're saying, and I totally agree with it.
now, if I could just give you a, let me see if I have this in. Um, so the creators are like, Sal, why do you believe in all that you said with the hierarchies and all? Why do you believe um, in common design? First, for those who've seen my Abiogenesis series and then James Tour series, um, I would argue that life is intelligently designed.
Right.
So that would be Michael Behe's viewpoint that life is intelligently designed. The question is there universal common ancestry? So one can believe in the special creation of the first life, but then um, common descent from that first life or that first pool of life. That's actually the view that was articulated in one of the versions of Origin of Species by Darwin. Whether he actually believed what he wrote has been
Right.
debated, but he talked about maybe all these creatures descended from the first ones that were created. He actually used the word created, which is kind of cute. Um,
Right.
it's somewhere there in Origin of Species where um, some people have argued that maybe he used that language to kind of just appease the other side. And, I would only Darwin would know why he
Right.
used that language, but he actually did phrase it in terms like maybe um, these first creatures were created and then
Yeah,
they
like breathed into existence or some phrase
yes,
equivalent to that.
right. So I could see how he used certain language that was a little bit ambiguous as to what he really believed. I
Right.
mean, he's trying to be
Like
diplomatic.
he only mentioned humans in the book like twice, I believe.
Yeah. So, oh, I didn't know that. So anyway, um, that's why I would endorse intelligent design. I thought James Tour made an outstanding
Right.
an outstanding chemical analysis of even though he didn't go out and endorse ID, everyone else, a lot of this ID sympathizers could totally were willing to go one step further and said, yeah, a lot of them I could tell you would not um, go to a naturalistic viewpoint
Right.
after hearing that. And um, uh, I could list the names of the biochemists, the organic chemists, the physical chemists, the synthetic chemists that have appeared on, albeit briefly, on YouTube channels to sympathize with both James Tour and also the view that at least the first life had to be designed. And I think that's a very strong case. So the problem then is for the young Earth creationists, it's like, we'll accept intelligent design, but how do we falsify universal common ancestry? And I say, don't try to do it all in one step.
Right.
Uh, uh, it's great that you had the abiogenesis. So let me see if I could find, if you had some thoughts while I look for my slideshows,
Okay.
um,
Yeah,
I'll try to
I
listen
guess
at the same time while I dig it up, and then we could have a talk.
right. Right. I guess on the subject of James Tour, I think you and Emery were responding to my comments the other day on LPP's channel on the relevance of expertise. Um, I think although maybe I'm just misinformed on how science works, which is a distinct possibility, but um, I think James Tour can say some relevant things on the subject, but I think that perhaps that work needs to be scrutinized even harder, if that makes sense. Um,
Oh, well, first off, I'm really glad now that I found out you're just a high school senior. I had actually assumed you were a college
right,
biology student. What I would just say is James Tour's audience was the ideal audience for his talk would be college students, but either chemistry or biology students. And I would just say we won't even talk. We After I saw that and he explained the theory, he basically made very basic textbook arguments. It didn't matter what his level of expertise was. His arguments were actually first rate.
Right.
And I felt that he did a great job of tearing down the origin of life researchers' claims. The experiments were correct, but what I would say, it's incorrect to say that you can take a 90-page recipe and of a chemical experiment and then try to represent that as what happened in a random soup. He made a very good case that was inappropriate. And he said a lot of these.
I think he was specifically attacking more the media of it, the media side of it. Am I wrong on that?
No, he was also attacking the researchers themselves that let this get out of hand, and then also both their abstracts and conclusions, which are saying this suggests a root. And it's like, okay, what do you mean suggests? Does that mean like it has a 0.0000001% chance of being true? That's not exactly representing the truth, because I worked through some of the numbers myself, and he worked through the experiment. So what I'll say is, there's no point in arguing this back and forth. When you get some more organic and biochemistry, and you actually look at his arguments, right. And I think he made a good case that this is not the reactions described by the origin of life researchers are very unnatural reactions. So much has to be forced to be able to make it happen. And so I would just say that probably at this stage, it wouldn't be productive to be arguing back and forth.
Right.
I will put stuff on the YouTube for those who have less chemistry background. And then also it's a chance for me to review my own chemistry. But suffice to say that there are a lot of organic chemists, synthetic chemists, biochemists, physical chemists that sided with Tour. They said we've worked on reactions like this. To represent this as happening in a prebiotic environment is really, really stretching it. This thing about contamination is a serious problem, because when you add contaminants, these things are so reactive that they would not result in the sort of chemistries that we'd see, even something like RNA or DNA. And he made very good cases with that. You don't need an expert. He was actually appealing to textbook chemistry at that point.
Right.
And that's one reason, I used to believe in evolutionary theory, and then was an old Earth creationist, then young Earth creationist. When I started applying textbook reasoning to some of the stuff that I actually heard that were in peer-reviewed papers, it seemed to be a disconnect. And I had the opportunity to come at it from both engineering and physics background, so I had a little bit more quantitative background to analyze some of the claims. And the longer that I had kept looking at it, that I was just like, I can't believe some of the stuff that's being put out there. So I would just say, Immutable says the arguments I've seen are poor. So, okay. So I'll just point that out. And Immutable, to be fair, it'd be great to have a discussion on this, because, on my side, I've had the physical and organic professors of physical and organic chemistry right here on this show talk about the problems, and they're qualified to make judgments like that. And this would be an interesting conversation to have. And it's not one that it's one that I feel would be worth addressing. And if Immutable is saying his arguments are poor, this would be worth a conversation that would be of interest to creationists and those
certainly not a conversation that I can put much insight into.
Well, but you'll have the opportunity in a few years. In a
That's
few
true.
years, you'll be able to see with the arguments. So you won't be in the dark for long, is
Right.
all I could say. You'll
Right.
be able to weigh in on this. And it's actually been interesting that at this level that we have professors on both sides having a discussion. It's not like, unfortunately, has been represented in the YouTube sphere where we have a professor versus Kent Hovind who doesn't actually understand the chemistry. We will have chemists of good quality on both sides making the argument. And so it should be interesting. Oh, apologies. If I get to come in, I'll explain why this is the case. Yeah, let me hang on here, Immutable. This is, I think I have.
Right. Yeah. Or I was just about to say, should we? Or it seems
Yeah,
like we've been going for at least an hour. So should we open this up for a short open discussion before we part
yeah.
ways?
Let me see if I have Immutable's email.
Okay.
I have it spread over accounts here,
Right. Yeah.
and
We don't want to let just anyone in here.
oh yeah, been having some vandalization. Let me see. I had some. Immutable, do you have a public email that I could use? Otherwise, I can look it up on one of my.
Oh, there we go.
Okay. I'm gonna.
Okay. He says give him a few minutes.
Okay. Yeah. I didn't mean to diss you there, Immutable. I respect you a lot. So.
Oh, Emery is still here. If you want to send him a link as well.
Yeah, Emery, you're welcome to. I'm gonna see if I need to get my sock accounts. I have to figure out which one I'm communicating from. So give me a moment.
Right. And I just wanted to say what I appreciate you letting me be on here on this channel and letting us talk, and of course on other channels too. It's it's often the case that many of the people you talk to are the most vocal people, where in reality sometimes the best people to talk to are the ones who actually want to have a conversation with you.
Yes, I
If
found that to be the case. Um, Andre USB.
How long have we been going, if I'm asked? Because I can't see a time, so just curious.
1 hour 45 minutes.
Okay.
And I think let me make sure I have Emery's email.
I guess since we've talked on phylogenetics a little bit, I mean, if you want to continue on that, uh, but I was also hoping we could talk a bit about some physics and any research you do in that area.
Sure. Okay. I just remembered Immutable had some problems using the Restream link, and if it fails, uh, Immutable, we can continue the conversation. So if it fails, apologies in advance for the technical thing.
Mm,
If they wonder why I'm being such a cheapskate, it's like, okay, I have Zoom accounts. I could get a StreamYard and then all these others. And it's just like, let me just figure out what I'm going to do, because that money could be expended elsewhere. And then also, too, I'm not so sure I want to be spending 50, 60 hours a month
right,
on the internet in live streams, unless it's something productive like actually teaching chemistry classes.
right.
So it's also to kind of put a check on me. So Andrew, Andre, Immutable. So we have Andrew, who is a rational empiricist, and Andy, who is the immutable destiny. So
Right.
Andy and Andrew. Okay, I'll try to.
I've been seeing a lot of Andrews around here, so it
Yeah,
gets kind of
yeah,
confusing.
yeah. So I think I have this. I have my. I wanted to show something first, and this is. I don't get a lot of disagreement on this, but let me see if I could. No, that's the wrong one. Promiscuous. I'm just. Let me close down some slideshows
Okay.
because I'm going to get so confused between them. And I really want to thank you for bearing with me, and I want to thank the audience that was looking into this nerd
Oh,
show,
it's not a problem at all. Actually, it gives me time to put my thoughts together.
because one thing I wanted to point out is, it's the creationists. They love YouTube, and I thank God that they do. Um, but if we're studying this at the college level, like you, the typical college student will be spending 15 hours in lectures a week.
Right.
And so a 3-hour lecture on this should not be just, that actually seems like not even doing any topic justice, because it's
Right.
only just like one small little thing. So,
I think that kind of gets into, and I don't know if you would agree with this, but like just going on, like and like I do this, but I don't claim to be an expert, for instance. But like some people who do go on YouTube and Google, for instance, and look at these subjects without any professional training, and do claim to be experts, I think they're in the wrong there.
yeah, yeah. Yeah, I mean, I don't think you need a formal degree, but I'm just like, sometimes I
Right.
just cringe because it's like I could tell you don't know this
Right. Right.
stuff.
It should ultimately be based on the arguments themselves, but sometimes the practical reasons credentials do matter. Would you agree?
Um, Sometimes, I wouldn't say always.
Right, right,
Like,
yeah, yeah. Sometimes,
yeah, it helps. It helps. It means
right,
at least the credentials will at least represent that you're willing to suffer through learning,
right,
uh,
right,
which really does count for a lot, because that's telling the world, hey, I suffered through this. I was willing to learn, and so some people were accusing me until I started going to school, back to school. Then they got really angry at me. It's like, oh my goodness, Sal's really serious. He's actually going back to school. So I got a master's in physics,
hmm.
and then I got the equivalent of a master's in biology. It's like, oh yeah, yeah, I don't have my PhD, but I certainly at least tried to study what you guys, what I'm trying
Right.
to talk about.
So,
So,
so, so, welcome to the stream, Memory, and also Dapper Dino is here in the chat.
yeah. Oh goodness.
Oh dear, the
um,
dinosaur is here. Thank you for letting me in, so I appreciate it.
oh, thank you. Oh my goodness. You've been active on YouTube today. So let me see. I'm going to share my screen here, and I'm just going to briefly talk about, because I'd like to. Let's see if I can get this. Were there some aspects of? So I showed all of this. I spent the last hour and a half saying, "Look at all these nested hierarchies I can build," and people will say, "Well, then why are you still a creationist?" Now I'm going to start to explain parts of it. So even, yeah, that's my professor there. That's Eugene Koonin. My professor was his staff worker. We eventually reached out to Koonin, and I hope I didn't get my professor in trouble with him. So yeah, I'm. Let's see here. Immutable, Andy. Good
Hey,
to see
hey,
you.
how's it going?
Hey,
Thank you for
Mutable.
joining.
Yeah, thanks. I appreciate the fact that the conversation so far has been really respectful, and I learned some stuff. Had a crash course on amino acids and things like that, and yeah, it's been a while. If you don't use information, you lose it.
Yeah, that's definitely true.
Yeah.
All right, so we have Andy. Why don't you tell the audience who you are a little bit? Your
Yeah.
biochemistry.
Right. So my name is Andy. I'm a biochem major. I got my major 2 years ago from local college. Mostly, I kind of, if it's science or philosophy, I really don't care. My interest is more so kind of analyzing the arguments people make and trying to address the argument itself, not address, not trying to attack the person. Speaking of which, Sal, I know that like on Speech Channel, occasionally we go after you and say some things that are not necessarily the nicest in the world, and I do hope you recognize that that's not like an insult towards you. It's kind of playing in the spirit of the theme of the channel. I know you and Speed have used in the past, so.
Well, I don't visit that channel, and Speed and I have had a mild falling out. I wouldn't say we hate each other or anything, but I just didn't feel it'd be a healthy relationship anymore. And I mean, I thank him for spending the time he did with me, but we have kind of a agreement, at least I said I'm not going to try to talk about whatever goes on in this channel very much on mine. I'm going to focus on like the discussions that A Rational Empiricist and I have had today. Just
yeah, I agree.
Not to talk about what other people had said about someone else. I don't want to get into creating drama. We will just talk science, and that's what I appreciated. So if you could, if it's okay, gentlemen, just for A Rational Empiricist's Andrew's sake, I'd just like to complete a few thoughts, and then we can open it up a little bit.
Sure.
So
Sure.
this is an example of the spelling of a zinc finger here. Captain Kirk, humanoid. All right, that's why I picked him. In the corresponding homologue in a pig, and you could see they look very similar. I took the trouble of highlighting the non-random pattern here, where you see the C's align, even in that protein, and then the H's. That's what they call a zinc finger. See if I can actually point to the zinc finger architecture. See if I have a picture of it here. It was actually in another slide, but the zinc finger, it creates a specific fold. They would even go so far as to say it's actually even a tertiary fold, because it kind of can exist by itself in the protein. And it's kind of like keys or locks, whatever metaphor applies. You can kind of see that they have a similar architecture, but they're not identical. And so these zinc fingers, each line is a separate zinc finger, but it is spelled differently, and that has a specific function that it helps connect to a specific part. So if we have multiple zinc fingers, just for illustration, we can actually, when we concatenate them together, they can identify a segment of DNA very precisely. It can look, an array of zinc fingers can identify a segment of DNA out of those 3 gigabase pairs. It's extremely precise, enabled if we have what we call a monovalent zinc finger. They're multivalent, that's another story. But they're you can either have it's just like locks and keys. You can have like a master key that can open many locks, or one key that's very, very specific that can only open one lock, or conversely a lock that is opened only by one key. So whatever metaphor works as metaphors go. So this zinc finger protein can then identify very specific regions. It will bind, it will connect to specific regions. So this is an example of the crab zinc finger protein, and one such crab zinc finger protein is this zinc finger 136, and we have one in humans. We also have them in pigs. And then we can take these and build phylogenetic diagrams, as you saw me do with the mega program, And then we also have collagen proteins, and it also has a non-random pattern that you could see here. The glycines repeat. That's very important for its biophysical characteristics. So I just happened to find these two proteins that happen to have, you can visually see the patterns that are non-random. And again, we can build phylogenetic diagrams. We have the human collagen type 1 and the zebrafish, and they're about 75% similar depending on the scoring methods. And you can also build phylogenetic trees with them. So it's like, oh, that superficially looks very powerful for evolution. But the challenge is you can't take, it's very hard to construct what the ancestor of a zinc finger and a collagen would be. And as far as I could tell, no one really disagrees with that. But I said, okay, that for creationists, this is already looking very much like their orchard model. Emery, do you have some thoughts on this?
Sorry, I actually clicked away for a second. Let me have a look at what you got up on the screen. I would largely agree. That does look an awful lot like an orchard model where you have different non-overlapping trees.
Right. And so this is where I would say we would have a problem with macroevolution of, like, say, okay, just to simplify it, this collagen is not going to evolve to be one of these because it's going to break its collagen pattern. Or if we tried to evolve this to a collagen, this zinc finger to a collagen, it would break. And by way of extension, it'd be very hard to imagine what the common ancestor would be that could be functional, because you don't have a collagen and a zinc finger ancestor that can do both or something similar. It's just not going to work. And so I asked Dr. Dan, CRISPR-Cas, both Immutable and Speed of Sound, and they said, yeah, they're cool with the idea of independent origins for the proteins. I said, okay, at least we have some sort of agreement that there's kind of an orchard-ish model. I mean, the creationists will say these are created. Andy, go ahead.
Yeah, there's something I should add too. So, like, what you're mentioning, the different colors that you have for this, I'm assuming it's an amino acid sequence, right? For either collagen or zinc finger, right?
Right.
So, something like basic enzymology for some people is that, look, this phenomenon shows up in basically all proteins. All proteins essentially require a handful of amino acids that need to have a specific geometry relative to one another in order to create an active site. But what I should add to that is that while your point is valid, Sal, I think if you're looking in terms of like common ancestry or things like that, or if what you're trying to do is use common ancestry, what you might be interested in doing is, instead of observing just the active sites, you're familiar like with various folds that occur in proteins, right? So like alpha helix, beta sheet, things like that, and the higher order structures that they form.
Oh, yes.
Right. So what I propose, right, as a way of kind of thinking about that is, first the question is, if you're making a protein, why bother having these higher order structures that proteins have? And to me, the answer is because certain properties that those residues adopt, like alpha helices or beta sheets, lend themselves to being more likely to be in some environments rather than others. So sometimes alpha helices are very, very polar, based upon the way that the side chains of the amino acids have to interact, and so you would expect it to be sometimes facing out of the membrane rather than facing in, where beta sheets tend to be more nonpolar in structure, if I'm recalling right. And the reason I point that out is, if what we're going to try and go on is evolution, right, you may want to consider evolution within a more broad context of, okay, it might not be possible for certain proteins to change amino acids in this particular kind of way, from zinc finger to, say, something like collagen, right? But what we might want to consider is, if there is a similar amino acid similarity, is there also similarity within the types of folds and domains that these proteins share? Right? That's just as important as a question.
Well, I'm not ruling out, just for the sake of argument, and obviously I'm a creationist and a young Earth creationist, I'm not ruling out that in the model of, like, say, Michael Behe, that, within, like, say, the collagen family, that one collagen could evolve to a different kind of collagen. And, I could grant that for the sake of argument. But what I would find very problematic is that the collagen and the zinc finger would have a common ancestor. And as far as I know, not even evolutionary biologists would even postulate that, because this crab zinc finger architecture, it's like, when I read the literature, it's almost like it came out of nowhere.
Yeah. I mean,
it started to appear in the vertebrates or something like that.
Yeah, there's some proteins. There's some proteins that are very strongly related to each other, but there's some that don't relate to each other in any way. And again, that's back to the point of the types of domains that certain proteins take on in terms of, like, secondary and tertiary structure, and also based upon the particular active site of the protein or regulatory domain that's being involved.
Well, I would say this: it just, for me, what is the mechanism that would be able to create something like de novo, an architecture like this? And it is acknowledged we would have to do cut and pasting of zinc fingers from another protein. You'd have to excise it exact positions, and then we have actually this crab domain here. So I'm just pointing out, if we're invoking natural mutation, random mutation, both point mutations and translocations, it'd have to be very, very fortuitous to make something like this. And this protein is not, as far as I know, really enzymatic. It's a connector protein.
Yeah.
And to show the complexity of it connecting, see if I had a picture of it. For it to be functional, first off, it needs to connect to a DNA, this particular one, and then it's connecting to other partners.
Right.
The odds, I mean, the reason that I have, and they say, oh, we can make de novo proteins. Let's grant that, because a tornado passing through a junkyard can probably make paperweights really easily. So there's certain kinds of classes of proteins that I would say, I could imagine a random mutation could make that. But then there are some that are so integrated into systems like this. It's like, to do this in gradual steps, just me at a personal level, I said that really just seems like a stretch.
Yeah, so I think it depends really on the type of thing you're talking about, right? I think everybody will agree, even people in evolution will say, look, there's an alphabet, or there's a lexicon of proteins that are kind of needed for life to survive and for life to function. Now, the real challenge, I mean, going to the origin of life or things like that, is really the question of determining what is a minimal genome? What does that look like? What kinds of structures are minimalistic for a first cell or something like that to be able to use and to survive on, right? Because once something works, like a zinc finger domain or something like that, there's no reason to really change it. There might be reason to add on to it, for the reason that once you add on to the domain, you may be able to do things like add regulatory functions or things of that nature. But there's no reason to really change the domain itself if it works.
I mean, I think I sort of get what you're saying, but I'm just pointing out to the viewers, this is why it's like, if I were Michael Behe, who accepted universal common ancestry, I could see why he would argue for intelligent design. This is the odds of getting these kinds of connections, when he's talking about binding site interactions with these zinc finger domains, if I could point out, for the monovalent zinc fingers, you'd need to get all these correct. Well, and we have experimentally tried to build these because we're trying to build artificial transcription factors. We're having it's a brutal engineering process. It's like, this is not, to try to do this with random mutation, we don't even try. But even with our best computational methods, we're having a hard time getting to program this zinc finger to be able to bind to the desired DNA. And that's just binding to the DNA. We're not even talking about all its other binding partners as I depict here. Go on, Andy.
Well, let me ask the following question, right? And this is another kind of question that's semi-open in the field, which is, do you think that the mechanism that this particular the way that this DNA is replicated by this process using zinc fingers is something that would have been present at the earliest phases of life?
I'm not making an origin of life argument here.
Well, I'm not saying origin of life. I'm saying once life exists, but in a very basic, but in a very basic form, like bacteria or something like that.
Can you repeat the question?
Yeah, so I'm asking this form of replicating DNA using zinc fingers that bind to these specific elements.
Actually, it's not replicating DNA. It is it's This actually happens in eukaryotes.
Okay. So the question is, okay.
Yeah, and it is for regulating. It's for regulating gene expression, but it's really complicated because these are in non-coding regions where it does the regulation on top of it.
Right. Yeah. Okay. So what I would say in that case is, like, I actually have a few books on my hard drive regarding post-translational modifications. From what I'm able to tell, and if I had to make a guess, I would say post-translational modifications are probably one of the last phases from an evolutionary standpoint that gets incorporated. And
ironically, I'd actually agree with you if we had the evolutionary model, and humans have tremendous amounts of post-translational modifications relative to other creatures based on the data that I've examined professionally, which is a very small sample size. But
yeah, the reason for that is like one of the processes you're referring to now. This process of it appears to, like, one of the things is UB, which stands for ubiquitin, and you can see there's, like, apparently something happening with a sulfur molecule or something like that. This kind of molecular action is very complicated and very and in many ways, like, very well orchestrated, very beautiful process, but it's very complicated. It's very hard to get molecules to kind of behave in these types of ways, and that's why some chemical reactions you have to play dirty and quick with it. Certain chemical reactions, not this one particularly, but what's required is usually, and you see this a lot in biochem, is you see that certain reactions will only occur once ATP is hydrolyzed, because the reaction itself takes a lot of energy, and you have to have a trade-off for a high energy reaction to take place. But so yeah, I mean, I think under an evolutionary model, because of, like, what I'm seeing here in terms of the what appears to be a very kind of like higher order and hierarchical regulation, probably not something that's in terms of, like, origins, probably not something that happens till, like, very late on.
Well, that's assuming that complexity can accumulate, but we have one of these rare situations where we agree that this is more complicated than what we see in bacteria, and I'd argue there's good evidence that, especially for like human beings, that this sort of thing is very much more complicated than the regulation we see inside bacteria.
Well, sure. I mean, the thing is fundamentally, though, a matter of regulation is a complex process, but from an evolutionary timescale, it's had a lot of time to play this game. It's had a lot of time to go through chemical pathways that fail, chemical pathways that succeed, and some that are kind of messy.
And that is actually a good question that's continued to be explored. John Sanford published on the waiting time problem that actually explores the difficulty of some of these things evolving in hominin populations. He actually was interested in this zinc finger because it is amenable to some degree about being able to calculate the time in co-evolving all the parts. And we just never got around to it. I said, John, I have other things to do. This is,
Well,
so let me just put that out there, that the problem that I had is that the evolutionary biologists would just say this evolved, and I said, you haven't done the due diligence to actually estimate the probability of this. The waiting time problem is known. That's not a term that, as far as I know, was created by creationists. It was an acknowledged problem even in their own literature. What you were saying, well, we had a lot of time to evolve this, and then when we actually get to the nuts and bolts of it, it becomes problematic. So I just haven't gotten around to looking into this.
So I'm not familiar with the exact problem, but I think I might be able to provide some insight. So here's the thing about natural selection and mutation that's kind of important to remember, right? You might have the best luck in the world, right, in terms of the types of mutations that accumulate and functional domains that are generated or things like that. But that's not entirely what evolution cares about, right? Evolution cares about a few things. It cares about the ability of an organism to reproduce. But furthermore, so even if you do have reproduction occurring, right, what's also important? The ability of an allele, the ability of some like new genic trait or some variant of the gene to be passed down within a population. And if that doesn't happen, that allele is gone for all intents and purposes, right? Because there's not an environmental pressure, there's not some pressure which is encouraging or favoring that particular variant to get passed down. So I think from where I'm coming from, and I'm not anywhere near as educated on some of the particulars as you, but one of the questions is not just a matter of saying how many step-by-step processes do we need to go from point A to point B, but what processes, what does history tell us, or what does history give us in terms of hints or clues that might have favored this particular evolutionary adaptation, or this new trait arising, kind of what would appear to be, in terms of geological scales, instantaneous or suddenly?
Well, as I said, just from an intuitive standpoint, and just as I saw this, now that I couldn't make the numbers work, because the numbers became astronomical. If I could just show the actual zinc finger here, that's a lot of letters to get right. We don't have to get every letter just right, but I was trying to do some counting estimates. I'm just like, each time I ran through this, I said, okay, if we have a monovalent zinc finger, this is going to be hard. And it's like for every letter that you add that has to be there in the right place, that's like adding a zero in your probability. And it just gets bigger and bigger. So, as I said, we didn't have a chance to formalize this, but even some of the waiting time problems for primate-like species that, have a certain reproduction rate, for them to develop new zinc fingers, when Dr. Sanford published on this, he wasn't specifically talking about zinc fingers. He's talking about transcription factors in general. And he said even just a few nucleotides would take hundreds of millions of years. And so when I showed him this zinc finger stuff, it kind of blew his mind. He said, it's not just a few nucleotides. You're getting into possibly hundreds of nucleotides having to align up. And we just never got to the point of working through it. I had other things to work on.
Yeah, I mean,
you actually saw one. You've actually seen some of the other projects, which is like the topoisomerase and the collagen project, and then now I'm doing stuff with bioinformatics. So it's like, there are not many creationists who are qualified to actually talk in these. We're really spread thin because we don't have a lot of grants to say, hey, God made this. We're not going to be able to get an NIH grant to do that.
So I'm not very familiar with this protein, so let me ask a question about it real quick.
Yeah.
So these different colors that are going on with the go back to the thing with multiple colors. Yeah. There you go.
Okay. So the color coding, I tried to reflect this Venn diagram here that you should recognize, but I didn't I didn't get the colors right. Okay, so that's the first.
No.
You
That's not what I'm asking about either. What I'm asking about kind of. Oh, there we go. What I was asking about is these blue sequences, like the different monocolored sequences you're referring to, right? These, so like the first line, like TGEKYDCKECG, yada 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 yada. That is a zinc finger domain, right? If I'm not misinterpreting the
Okay.
protein sequence.
The signature of the zinc finger, and unfortunately I don't have the diagram with me, is you want to have these C's, these cysteines here. It's separated by 2 amino acids, and then some distance further you have the histidines, these H's here. That's the signature.
Okay. Yeah, I was kind of wondering like what all these colors are going around, because if it was all these things needing to interact, I'm thinking, yeah, that's kind of, yeah, that's a tall order. But a more general question, because I don't want to get too caught in the weeds with biochemistry, because I could do it for days and just lose track of time. Is would you agree with the conjecture, with the kind of like the adage that if the zinc finger domain or motif, or however you want to refer to it, is something that's ubiquitous and that change or appears in multiple forms in life, do you think that kind of renders itself as favorable to the idea that in some sense it's easy to evolve? Whereas when you compare like other protein domains, it might be the case that you can't have any mutation or very minimal mutation occurring, like within, I think you mentioned like histone proteins.
I would say that I've done some statistics. Making an individual zinc finger, getting the histidines and the cysteines in the right location, I would grant that that's feasible. A single zinc finger, but to get one that is optimized to target a specific region of DNA or whatever its binding partner is, that is not so trivial. But then the other thing is, a lot of these, the reason you see it as an array here from a functional standpoint is one could think that, I'm sorry, I'm clicking around here. I can't click and talk at the same time.
Yeah.
Is that an individual zinc finger might be able to target, like, say, 3 nucleotides, and that, you don't get any specificity with that. To be able to get it to go to the right spot, you really want it to target a longer stretch to be a good transcription. Some of these are transcription factors, but some are not. So for it to be functional, that's where the waiting time problem comes in. It's like we can evolve a zinc finger, but is it usable? It's just like saying, I can make a key, I can kind of hammer it out. Do I have a lock that it corresponds to that makes it usable? And that's actually the statistical problem. We can evolve it just like we can put an insulin inside a bacteria, but the bacteria has no use for it. And so the problem of the probability of it evolving is really, are we able to evolve it in a way that it has a functional context within that organism? And that's the real probability problem, not just making something with it could hypothetically, we could take 32 amino acids and say, what's the probability we could eventually get a cysteine here and a cysteine there and a histidine there. That's not too astronomically remote, in my opinion. But being able to get an array here, and that's been my criticism of some of the evolutionary literature, that where it could target this, and then also more problematic is, let's see if I can have a picture of it. I had that picture of the, this one, where we have the retrovirus changing, and then you happen to have a zinc finger that's going to target it, and they invoke coevolution. And then as I studied the construction of artificial zinc fingers, a little perturbation like that is not easy to correct. It's not like you have gradual ways of tweaking it. It tends to be like a password. Unfortunately, I don't have enough background to be definitive on that. Go on, Andy.
I think there's something critical you just mentioned that's that might help answer the question inadvertently. You said, or you seem to imply, like that the system is really sensitive to perturbation. What that means for people that's not really too familiar with what that term means, it means that it's very sensitive to being touched, to being changed, to being altered in some way. Um, from my familiarity, and I could be wrong, right? And feel free to interrupt me and just smack me if I am. Um, Viral elements, at least certain viral elements like tandem repeats and things like that, are very short. Um, are very short when they insert, and what might explain something like the need for a zinc finger protein to be developed, or to, kind of, as you're saying, consistently target segments of DNA, might be the fact that there's some entity that's trying to replicate very specific small fragments of DNA, in very discrete chunks that's easy for a protein to recognize, right? Like with viruses. If a virus replicates and it's harmful to its host, it's going to kill its host, and you won't have the genetic material from that cell passed down. So domains that are able to bind to that area and that are able to repress transcriptional activity aren't able to be passed down.
Well, let me say this, that I think we can tentatively agree that this question is worth exploring
Yeah.
more carefully. And obviously I have my biases, but
So do I. I don't think
when I share this with my colleagues, they're like, yeah, wow, there's just no way. And it's like, okay, I recognize that that's an intuitive thing. If we want to be more rigorous about this, we'd actually start to put it through some more careful analysis and then try to publish. We just haven't gotten around to it. And then the other side will say, oh, this easily evolved. Of course, we would complain about that.
Yeah,
but I'm glad to at least talk about the chemical architecture here, because
I think certain things. Yeah, I think certain things you say, like it's sensitive to perturbation. Like when I hear that, right, my mind is very analytical. So when you say something like it's very sensitive to perturbation, what I'm thinking of is, okay, what kind of process, or what kind of thing, would be sensitive to perturbation? Because like you've explained so far, and I'm by no means an expert, so I have no authority to really speculate on this. There's very specific areas where it needs to bind, and those regions, are sensitive to change. And so, to me, telling me that it's perturbative to, that if you perturb the system, it's very fragile to that, seems to tell you something about the problem, or something about something potentially, at least, about how this thing could have evolved if you're accepting evolutionary framework as a model to explain.
I think you characterized it correctly, that this is, and I just for the sake of those who really are familiar with zinc fingers, we had the monovalent that bind only specifically to one, and then the multivalent that can bind. There's like the CTCF zinc finger. It can bind to all sorts of things. So I'm specifically talking about the monovalent ones. And there is one particular one, the transcription factor, III transcription factor 3A, TF3A. I know that these proteins have these strange names that only just like these little...
You might as well be speaking Latin to me, Sal. Unfortunately, I'm not a bioinformatician, so I can't just pull out of the lottery and tell you.
But where I was headed with that is definitely one that even the people who reported on it were just amazed at, because it was very specific to the DNA. The zinc finger would interact with I have it right here, segment of 5S DNA. 5S is like a ribosomal RNA or something.
Okay.
It codes for it. Okay. So it specifically will bind. This is like 100, anywhere from 50 to 100 nucleotides. It's very, very specific.
Yeah.
But the amazing thing is it would also bind to the RNA using a different mechanism. And that's what I mean, very sensitive to perturbation, how it did that. I mean, even in the literature, they're just like, wow. So I know this is deep in the weeds, but
No, it's not my intention to get too deep into the weeds because, like
Well-
I said, it'd be a perpetual tennis game going back and forth.
yeah, but if people ask me why do I think there's design, it's when I see this, because the literature says this is elegant, this is sophisticated, this is artful. We get those adjectives in these professional papers. I mean, that's how struck the researchers are at the extreme architecture. I guess they won't use the word design, but they'll say that this is very, very sophisticated in wonderment. And the reason that they'll say that is we're trying to make artificial systems that kind of do the same. We can't do it as well as what we see inside living organisms. And I'm just trying to communicate that intuition. It's kind of great. We're trying to do genetic engineering to at least teach ourselves and to further medical science. But in the process, we've begun to see like how clumsy some of our efforts are compared to the way that we see it already implemented in biology. So this TF3A zinc finger protein that is able to bind to a segment like this, the researchers they're just in awe of what it can do, and they don't completely understand why it's able to do it so well. But
Hey, I've told people this before. When I was an undergrad taking like biochem 2, I was amazed at that like some of the processes work at all, because it's people that remember things like Krebs cycle or fatty acid synthesis and degradation pathways, stuff like that. There's so much going on there, and when you really zoom out at the picture, that's a process that's occurring in addition to hundreds of thousands of other processes in a cell at a time. And a cell is very much on a clock, right? There's times where it needs to grow, there's times where it needs to begin producing proteins that help it to divide and pass down its genetic material. And also, like you all know, it has to at the same time make sure that certain elements of DNA are protected while other elements are able to be transcribed. It's a mess. It's a very messy system. Let's put it this way: if it was designed, it was definitely designed by committee.
Well, yeah. See, that's why we get along, is because you study biochemistry, and it's something that I liked, and I'm actually now trying to reteach myself, because it's been embarrassed to say this 30 years ago since I studied general chemistry. So
trust me, I've only been out of the field for like two years now, and I'm going back for certification in biotechnology. So I'm going to run into this like face first against the wall. But even just being like a year or so out of it, it's difficult trying to recall some of the particulars regarding replication or some of the things you're talking about.
Or even general chemistry. I had to do something with significant figures again. I'm just like, oh my goodness, this is embarrassing. I had to some of it I know intuitively, but then to try to teach it, you're like, yeah, I think when you actually try to teach it and articulate it, then you become a lot more rigorous in the way you do things. And
I'll say this much. I mean, I think chemistry is it's a very Goldilocks science, and what I mean by that is it's not as mathematical as physics, and it's not as soft as biology can be. There's a nice kind of like lukewarm little area where you can play around with chemicals, be it analyzing complex physical structures like proteins, or understand physical interactions between molecules. But then you can also do things that James Tour does, like synthetic chemistry. And God, I have the highest respect for those people, and that is a monster of a field to get into, because trying to manipulate chemicals to react in a certain way is far from a simple endeavor. Yes.
Godzilla Freak asked when I'm going back to the Academy series. I've been meaning to do that, and maybe for good reasons. I've been distracted because the James Tour series got me thinking, and then of late I've been getting recruited to do debates and stuff. And I've been also branching out into a little bit of teaching the Bible and theology and political commentary. But how about next week? I'll get back into the series, God willing, and you can be praying also for my mother's health. I've been distracted trying to keep her alive. I mean, literally. I actually, even as recently as 2 weeks ago, I really wasn't sure she was going to make it. So it's been a very difficult time for me emotionally.
No, that's rough. I hope.
Yeah.
Hopefully
So let me just explain to the viewers, and I appreciate all their prayers and thoughts. When someone gets to be that age, a lot of things can go wrong. And then even little things that go wrong can cause someone just like to not even want to eat and drink. And once you stop drinking, you probably won't last more than a week. And right now, the biggest barrier is, her vitals are looking good, but she's sleeping all the time and not willing to drink. I mean,
Right.
I had to practically scream at her to wake her up, which I hate screaming at my own mother, but telling her you have to drink something now, and to get her to swallow pills is very difficult. And so a little bit of deterioration in her ability to do even something as simple as swallowing liquid, she won't be around very long. So just to share that. Yeah, thanks. Thank you. I appreciate the sympathies. So
Yeah. Do you want to go into the James tour thing now, or maybe we can make that its own conversation? Because
maybe
I did.
Yeah. Why don't we make that another conversation? Because we're already going on 2 hours and 16 minutes, and
I brought that up simply because it kind of showed up in the chat kind of as a natural course. But
yeah,
honestly, abio is as everybody knows, it's a very complex field. I have my takes of James Tour. I'm not going to challenge him on the chemistry, but in summary, I think the way he's thinking about it, but not just him. I think a lot of people are thinking about the field regarding abiogenesis and future research to be done on it is just incorrect, and that it's not productive.
This would be a productive conversation to have, and
that's kind of like the 10-second version of my view, but obviously at some later point we'll have time to go into it more carefully.
I'm gonna look. I just maybe to complete the show. So we can maybe say that we'll save that discussion for later. And I have your email.
Yep, you do.
And I'm looking for, just to close out. While you, does anyone have some thoughts while I go poking through my files? Because I want to close out something for Andrew before we say we call it a night.
Yeah, I do want to give you perhaps a constructive criticism and maybe a new way to attack evolution, if you don't mind.
Oh, go ahead.
Yeah, so let me say this, and my community is going to lynch me for saying this one. Evolution, to my perspective, it's a great theory. It explains some things, but there's questions made about how predictive it really can be. This is why things like extended evolutionary synthesis is something that's being pushed, because even though we understand parts of evolution and we understand organisms have an effect on their environments have an effect on their organisms, things like that, the dynamical process of that is still very much a mystery, and it's very much a mystery regarding the question about how that interaction unfolds and ultimately what's the long-term behavior and like as it concerns mutation or niche construction or things like that. I think there's a lot of strong ways to attack evolution, because evolution, although it's a theory that I think functions, there's room for it to falter, and there's room for us to improve it. And so the way that you may attack it is thinking about evolution, or what we call evolution, is a more holistic process, trying to figure out how various parts of this evolutionary ensemble are able to interact in a way, that makes sense. Because, like me and you, our educational background mostly regards biology and biochemistry. So to me, I can come up with excuses or come up with rationalizations on how a protein works or things like that. But when you ask me about things like development, or how ecological interactions take place, or how that subsequently has an effect on organismal fitness and evolution through the next 100 years, I don't know. Um, that's not a prediction that's easy to generate, if not impossible to generate. But that is something we should be able to do with evolution. We should be able to project it into the future, in terms of like the predictions. So I do think that's a realm where you can attack it on.
Yes, and it's kind of a sad thing. The early predictions of a creationist is that our genomes and the genomes of other creatures are deteriorating. Michael Behe, who is a biochemist, he published the book Darwin Devolves, and that does lead to testable predictions for eukaryotes. Bacteria replicate so quickly. I don't know that it's quite so easy. So the trajectory of evolution for us, let's just say hypothetically, the species is around for another 5,000 years. We might have a different perspective on human evolution, and then they'll say, well, we have a very large population. Well, we can look at primates or whatever.
Mm-hmm.
We might have some model organisms that are eukaryotic, multicellular. That could lead to testable predictions, which, personally, I think it will favor the creationist model. That's one reason that I'm a creationist now. I just, as I was showing all these complex systems, I said, there are just so many ways that this could go wrong. And, we were talking about the sensitivity to perturbation, which really says the sensitivity to mutation, of random mutation. It can really matriculate through systems like humans that are so, the pieces are very deeply integrated. Whereas I think bacteria, the mutations there are a little bit more forgiving.
What's weird about that, though, too, is it like when you consider a lot of cells undergo quite a bit of mutation, and there's some that are bad. There's some that are like really, really bad, and you don't want to have propagate throughout the genome. But the ones that really spell disaster are the ones that are that pertain to cancer. Once you begin messing with genes that start mucking up regulatory pathways and things of that nature. But other than that, there's I agree, right, that there's certain parts of proteins or certain things you just can't change. But for the most part, it seems, to me at least, right, and this is just my opinion, it seems that life has done fairly well in adapting to dealing with mutations and trying to kind of develop error correcting mechanisms for that, for this kind of uphill battle.
We have experimental evidence that there is a level of robustness. Brenda Andrews did experiments where she was knocking out genes, and she was actually shocked at her results. These single knockout experiments, she couldn't believe how many single knockouts she could accomplish in a yeast. Granted, that's not
not exactly representative,
but she was still like, wow, hardly any change in reproductive fitness. Once she started doing double knockouts, it became evident that you can't, you can. It's like spare tires. One can act like a spare tire if you knock out one, but you can't knock out both. And so you do cite an interesting property that there could be mutations to the system that are not immediately detected, and that also leads to a whole nother topic of deterioration. Then if natural selection is not able to arrest these very small, what we would call slightly deleterious mutations, then these deleterious mutations can accumulate, and that is still a very strong concern for human health.
Yeah, so there's
ironically, that's why they invited a young Earth creationist like John Sanford to speak at the NIH. There was sympathy for that view. That
there's a little paradigm shift with that as well, though, which is this gene eye view of the cell or of organisms. This idea that you have a gene, it does a thing, that thing does a thing for a cell, so on and so forth. It's worked so far, but one of the things that becomes really predominant when you study like cell biology and when you study processes within cells is, while it's important that proteins have the function they do and perform whatever it is they're doing, what's also important is how genes are placed relative to one another, right? So genes, in some sense, compete for activity on various processes. It could be that you have one gene that's responsible for fatty acid synthesis, but a gene, several thousand nucleotides, upstream from it does something completely unrelated. But once you activate the second gene, the first one deactivates, right? How there's this kind of very interesting juggling act between genes that activate but end up consequently temporarily deactivating other genes, and how even when you accumulate damage to one portion of a gene, or one gene particularly, there's other maneuvers that cells can use to kind of avoid this kind of catastrophe scenario that you're describing.
I'm just writing a letter to apologize to Standing for Truth. When I scheduled this, in my mind, I actually thought that Sal Giardina was speaking at 4 p.m. So I said, "Oh, I can have my show at 8 p.m." And then by the time I set this stream up with Andrew coming here, I already set it up. By the time I realized that I had a schedule conflict, so my sincere apologies. I didn't mean for us to be scheduled at the same time. That's totally on me. I have one last thing before we close the stream. I was going to share something. So I shared earlier why I think that it's problematic to have a universal ancestor for all proteins or / genes. And I don't think in general, I've gotten a lot of complaints about that theory. What I had a show, and we can talk about this some more, where we covered eukaryotic evolution, and we had Dr. Change Tan on, and also Dr. James Carter. And Change Tan was the lead author of this book, and she wrote some papers. I meant to say, I actually shared this paper on the problems with eukaryotic evolution with a theistic evolutionist. He was a Christian pre-med student. He became a creationist after I shared that with this individual. So this was a phylogenetic diagram. Earlier in the show, I showed that how we can use MEGA to build phylogenetic trees. I built this phylogenetic tree on the topoisomerase enzyme. There is a homolog that's a remote homolog in bacteria. It's a remote homolog. And thus I included it in the tree here. And I don't know if I went through. No, I don't have a picture of the similarities. But so this is a situation we can build a phylogenetic tree, and where I have gotten in some sharp disagreements with evolutionary biologists, saying that you can't use this to say that it evolved naturally. And it was in this talk. This is a slideshow from the video on this. This is some extraneous stuff here. Let me get to the heart of the issue. Okay, so this is the mega alignment between humans and bacteria. And okay, this is one thing of interest. The bacteria, what it doesn't have is a sequence like this here. This is a localization sequence. It's called a bipartite nuclear localization sequence. So I highlighted in red here where it resides on the human, and it doesn't, the bacteria have amino acids there, but it's not this sequence highlighted in red. What's the importance of this sequence? This sequence resides somewhere physically out here on the topoisomerase. This is the fold again. That's Dr. James Carter, not Dr. Robert Carter. That localization sequence tells the it tells if the topoisomerase is somewhere out here after it's been translated from the mRNA, it tells it to go inside. It needs the sequence to go inside the nuclear, the nucleus. So this sequence is very important. If we suddenly have a eukaryote that has nuclear membranes and a nucleus, it needs a way of having the proteins go from the cytoplasm back into the nucleus. And when Dr. Dan appeared on my channel, he said, well, you need to include natural selection in your model, Sal. He did see I'm inferring from his comments that he could see the challenge of evolving that localization sequence. And again, just for the reader's sake, this is the localization sequence right there, to evolve it by random mutation. And he said, you need to include natural selection in your calculation. Otherwise, I'm not interested. I didn't have a chance to respond because I saw his comment kind of only after the stream was over. And you know how it is in a live stream. I wanted to say, well, natural selection would select against this because if you get it half right, the creature's dead already. It's dead on arrival. And that's only one of the critical proteins. We would have to reformat all the homologous proteins. And so that was where I said, being able to build phylogenetic reconstructions is illegitimate to be able to point out these mechanistic barriers. So it's like, yeah, you have these nested hierarchies, but this is proof kind of in the Behe-esque sort of viewpoint that you would need fortuitous events or intelligent design. And of course, he'll say intelligent design, that even under the assumption of universal common ancestry, there would have to be these very fortuitous mutations. Otherwise, it's just not going to evolve. And
yeah,
go on.
Well, one I'll say about that is, I think if we start from any given protein, you want to consider, be it ATP synthase, be it the flagella or whatever, there's complexities and intricacies within the cell that are hard to account for naturally. That's a concession I can make. That's not controversial. In terms of what you're asking, right? In terms of this kind of process of reconstruction to find out, how likely is it for it to have evolved? I think we could probably agree for sake of argument that what we would want to try to do is we would see how well this protein relates to other proteins we might find. Essentially try to do some kind of multifactorial analysis in regards to trying to extract some kind of relationship or correlation relationship between certain proteins, or are protein 1 and protein 2 very highly related to each other? right, in terms of this common activity that we're referring to? And if so, right, if you keep seeing this pattern, then yeah, I can have the argument that that something like irreducible complexity, or something like it's not possible for this protein to evolve, while these other proteins need to evolve in a very particular kind of way. Yeah, that's a fair concession. I think just picking out one example, though, it doesn't. I'm not sure it tells you enough about. I'm not sure it tells you enough about the possibility, right? Because my main concern is that life, in all aspects, is a dynamic system. So everything feeds back on everything else, and what we should be considering is the weighting effect of variables that are relevant, and seeing how strongly something correlates with something else to try and base probability on those.
Well, I appreciate those sentiments, and where my complaint is that I feel that the evolutionary community should be addressing these questions too. Rather than just using a phylogenetic tree to say, hey, this proves it evolves, and they don't look at the problems that I've posed. That's the issue I've had, is that notwithstanding what you just pointed out, to make my argument more complete is to explore more proteins. The thing that I've seen with the evolutionary community, they don't even look at this protein. And it's like, before they start invoking these phylogenetic reconstructions, they need to look into these considerations. Now, in their defense, not that I am eager to defend evolutionary biologists, but I want to be fair here. We have a lot more data now that they probably weren't aware of in terms of the importance of specific sequences and their role inside the cell. We're only learning a lot. So there are probably questions that they had just assumed that would be easily solved that are now emerging that they have to consider. And I just, I'm now just kind of getting in their face and saying, hey, this is problematic. If we're trying to make these arguments at the college level to students, this is what I would say. I'd say, okay, we can build these nested hierarchies. I have no problem with that. But this is my issue, that it's not a proof that it really is feasible. Because when we do these, when Nathaniel Jeanson and Robert Carter do this using the same software with Mega, they're doing it within species, within individuals we know can reproduce with each other, like humans. Right. And so, yeah, we can accept random mutation as an explanation. Random mutation and natural selection is an explanation for the differences among the genes, or really alleles of the genes within humans. But it's trying to extrapolate that to something like the eukaryotic transition. They would have to deal with these sort of challenges. And that's where I had a fairly sharp disagreement with Dr. Cardinell, Dr. Stern Cardinell, is that, I'll grant that, because even creationists will grant that for species that are known to reproduce with each other. We have no problem with that. But to extrapolate those examples all the way back to eukaryotes is problematic for reasons like I pointed out. And of course they'll say, okay, well then let's just move on from eukaryotes. Let's look at other creatures. And that's fair game. That's fair game.
Yeah. So I think there's a few ways you could address this. I think you can do research in terms of evolvability experiments, trying to see how easy it is to get this type of protein with this function to kind of emerge. But in fairness to you, right? I do think that's a challenge for evolution. I think one of the great challenges of evolution, because it does apply to evolution, right, is this transition between kind of this, like, this LUCA, this last universal common ancestor of life, to eukaryotes. Because that, even according to evolutionary theory, that's an event that took over 2 billion years to occur. So this is not a common event. This is a very long and arduous process to get off the ground. So there's things you can do in regards to that, but there's also, to some extent, I think what both of us have to kind of come to terms with is that, number one, well, this is the only point. So, there's a kind of haziness regarding how much we can know about the transition from proto-eukaryotic life to eukaryotic life itself. It could be that proto-eukaryotic life had the ability to undergo some variant of horizontal gene transfer and then lost it for whatever reason, right? If that's the case, you could see the origin of novel traits like these to be very easy, because horizontal gene transfer is just a miraculous it's a miraculous tool for spreading about genetic diversity. But contrasting that, if you don't have some primitive horizontal version of horizontal gene transfer that gets damaged or just completely wiped out, well, then it is much harder, right? So there's a million and one possibilities under the sun for us to think about in terms of plausibility and how easy it is for some of these proteins to evolve. And there's research, I think, that that's kind of trying to parse at that question. That's trying to parse at why do certain codes within a DNA or certain codes that relate to amino acid sequences, why did they evolve? Why did they appear? And at best, we're going to have a probabilistic answer. But so, I do hope the community accepts your challenge, and I do hope that we, no matter what the result is, we find to some extent a satisfactory conclusion, even if we don't know what that will look like quite yet.
Oh, well, thank you for your thoughts. I'd like to, so I had pretty much covered kind of some of the things I wanted to share with A Rational Empiricist. He probably got way more than he bargained for in this discussion. But for a senior in high school, if you could even digest even a little bit of this, I totally respect you. And I did not know you were just a high school senior. He came across as being so knowledgeable. I presumed he was already a college junior or something. So hopefully I've given some ideas to think about, or at least areas to learn.
So in other words, Andrew, welcome to the nerd club.
Yeah, welcome.
Sorry, but I am already king of the nerd clubs at this point.
So, gentlemen, I'll just Emery, I'll start with you, and then Immutable, and then Andrew. Some final comments, and then we'll shut the stream down. I'd like to thank everyone for who's participated in the stream. And if you like our nerd discussions, please let your other nerd friends in and let them know about our channel. So, Emery, some thoughts?
I don't think I said anything other than hello since I came in the door, and the little cheeky comment I just made to Andrew. But honestly, I got a lot out of it from just listening to the back and forth. I thought that it was very helpful. I don't necessarily agree with everything that was said from either side, to be very honest, but I've learned a lot, and I think it was very helpful. And I think the discussion shows that it is possible, even on hypercharged issues like this, to have a reasonable, intelligent, adult conversation that does not descend into name calling.
Yep.
Well, thank you, Emery. And Andy, Immutable.
Yeah, man, it's actually been a while since I hopped on one of these streams. It's been very enjoyable. As always, I appreciate having conversations with people that don't necessarily agree with my position or don't agree with my position at all, because honestly, the only way you find out weaknesses with your position is by engaging with people that disagree with it. And there are things to be said in acknowledging holes or problems with evolutionary theory and trying to find solutions to rectify that. And we'll see which model comes out at the end. But as always, I always enjoy being able to hop onto a stream, and especially with a person like Sal, who I respect a lot, to be able to have a nice cordial conversation where, even if we don't agree on the vast majority of topics that we encounter, at the very least, there's a degree of rationality and intelligibility to the types of objections we make, and a productive discourse, hopefully, for somebody.
Oh, Thank you, Andy. And our guest of honor tonight, Andrew Cumming, A Rational Empiricist. I'll give you the final word.
Yeah, so just to kind of reiterate what Immutable said, I think that we're all human beings and we all have our biases and we all have things that are built into us, and sometimes it can be hard to cope with those parts, and I by no means am an exception of that. But like, I think that this discussion and many other discussions, and this is one thing I appreciate about you, Sal, is that we can have rational discussions, and we can actually kind of not completely, but turn the channel into a kind of rigorous or more rigorous and more scientific approach at kind of debate topics and things like that. And so I appreciate that. And so thanks for having me on.
And thank you all. And tomorrow, just as an announcement, S.J. Thomason will be co-hosting something with me at 2 p.m. on apologetics. We'll be covering some scriptures. And again, apologies to Standing for Truth. I totally messed up scheduling. I thought Sal Giardina was presenting at 4, and I scheduled this at 8 p.m., which I found out too late. And sincere apologies. I didn't mean to conflict. So thank you to all the viewers who've been with us through this 2-hour, 46-minute discussion. And thank you to all the guests here joining the panel. Take care. God bless you, and have a wonderful evening.
Keep being curious.